Hi, my name's Steve Chamberlin. I'm a naturopath and a postdoc at the Oregon Health and Science University. For the last year, I've been studying the effects of a water extract of the plant Centella asiatica in mouse primary cortical neurons. I've been doing this in the Department of Neurology on a project called Botanicals Enhancing Neurological and Functional Aging, BENFRA. The broader goal of this project, BENFRA, is to establish a pipeline to study botanical extracts for human health, and with the idea of taking them to clinical trials and FDA approval. This includes, extensive evaluation of compounds that are in the plant, biologically active compounds, and the mechanisms of that activity in those compounds in both in vivo and in vitro studies. This plant, Centella asiatica, also goes by the name Gotu kola, and it has a long traditional history of use, medicinal use in Ayurvedic medicine for enhancing cognition. And we've also seen that in our in vivo and in vitro studies or in vivo studies. We've seen enhanced cognition in the lab. We've seen with mice, we've also seen increased dendritic arborization in neurons. We've seen increased mitochondrial function and also activation of endogenous antioxidant pathways in the experiments that we've been doing. So a lot of these effects are thought to be related to several classes of compounds. One class are triterpenes, and there are 4 compounds we've been looking at, and then another class are caffeoquinic acids. There are 8 compounds there. So those have been studied a lot, but the purpose of this analysis is to do an untargeted transcriptomic analysis to see what are the effects of these compounds, what are the interactions between these 2 categories of compounds, but also are there other compounds, what other compounds beyond these are affecting gene regulation at this point, or gene expression. So the way that we set this experiment up, we use it's mouse primary cortical neurons. So these come from embryos, mouse embryos. We culture those for 5 days, and then we treat them for 48 hours with the different compounds of interest. And the way that we set this experiment up, we had a control, and then we had 4 treatments. One of the treatments was a full water extract of Centella asiatica. And then we had a treatment, we treated some cells with just the 4 triterpene compounds and some cells with just the caffeoquinic acid, the 8 caffeoquinic acid compounds. And then we had a treatment where we had both the triterpenes and caffeoquinic acids combined, and we're trying to look at interactions. One area of interest when doing a whole plant study like this is to try to assess any interactions, potentially positive interactions between the compounds for their health effects. So we have, by having a separate triterpene and caffeoquinic acid treatment group and then a combined, we can look at the interactions between those two groups and classify the interaction as synergistic, additive, or antagonistic, or inhibitory. So our results, you can see on the bottom, there are 3 areas of results. We're focusing in on sort of one function that sort of jumped out at us. There are a lot of other things going on with this analysis, but for this poster, we're going to just focus on kind of one area. So then the first on the left, the first box on the bottom left, is the differential gene expression. So each one of those 4, these are 4 volcano plots. Each one is for each treatment, and this is all relative to control. And so we have, on the y-axis, we have the p-value. This is false discovery rate adjusted p-value. We use 0.05 as a cutoff. And then we have the fold change between, the treatment, the expression fold change between the treatment and the control. And we're using a fold change of 2 as our cutoff, 2 or more. So all of the red dots are genes that meet both of those dimensions of both of those cutoff dimensions. So the first thing to point out here is that we did get activation in all 4 of these. We're going to focus on the upregulated genes with the full CAW water extract. So that's the arrow is coming from that. There's a shaded square around those genes. And then in the middle, so the next thing that I did was a pathway enrichment with the Reactome pathway database. What this is the detail is not really what I'm focusing on here, but this is all the pathways in the Reactome pathway database. The figure in the middle, figure 2. So that and then the size of the polygon is the number of genes in that pathway. So this is overrepresentation analysis, hypergeometric tests. So the where it's yellow, those are pathways that are enriched with the upregulated genes from the CAW treatment, and there are 946 of those genes, 946 genes. Yep. So so there you can see they're kind of distributed throughout the different functions, except down in the lower right-hand corner we have that's extracellular matrix organization. Almost every pathway in that category. This is Reactome is hierarchical in its organization, and almost every pathway is upregulated, and that is not an area that we have studied so far. But when you look at the literature, the other traditional use and current use for Cortical or Centella asiatica is wound healing and other skin conditions, and it's very well studied for upregulating these pathways, and especially collagen pathways. So there is some evidence that this can be neurologically beneficial. So this could be a new mechanism for the benefit or for the effect of this herb of Centella asiatica. And then on the right, there's a bottom right compound interaction. So this is a heat map, and the color is in the broad columns. So there on the right there are 4 wide columns. This is the heat map itself. So there are blue is low expression and red is high expression, and this is expression relative to control. It's not full change, it's just the expression value, the expression difference, the coefficient from the equation. So what we're trying to do here, this is an interaction figure. So we have the column for the so these are the extracellular matrix genes. So these are the genes that were used to enrich those ECM pathways. They're all significantly upregulated with the CAW treatment, and each column is their expression level relative to control for the 4 different treatments. So you can see on the left we start with triterpene, CQA, and then we go to the combined, and then we go to the full CAW extract. So and then on the left we have a couple of columns which give their differential expression status for each one of those genes in the individual treatments, and then there's kind of an orange and green column which gives the interaction status. So the bottom line here is that most of these genes are not differentially expressed with triterpene and caffeoylquinic acid. They almost half of them have a synergistic interaction when you put the 2 compounds together, and then they all have we see an increase in expression for all of them coming from the unknown compounds when you look at the full CAW. So this is kind of showing somewhat of a stepped up interaction as you move towards the full CAW extract. So if this is a beneficial action, of this herb, it's beneficial to use the whole plant rather than trying to isolate a single compounds for this. So there are lots of other, functions that we are seeing with this. This was specifically focused on one kind of interesting thing that jumped out with this extracellular matrix. We're also doing metabolomics for this. We'll integrate those and ultimately go into these potentially new mechanisms further. Okay, thank you.